Hello, welcome to our video where we will be teaching you about everything we know about DNA. We will talk a bit about how DNA was discovered, the physical components of DNA, the chemical structure of DNA, and the classification of the 4 nitrogenous bases. So, first a bit about how DNA was first discovered. There were many scientists involved in the discovery. In the US, there was Linus Pauling and Aaron Chargaff in 1948 and 1949. And in the UK, there was Rosalind Franklin and Maurice Wilkins in King's College London and James Watson and Francis Crick in Cambridge in 1953. In 1948 in the US, Linus Pauling made the discovery of the alpha helix whilst he was sick in bed. He realised polypeptide chains folded and formed hydrogen bonds. This formed a spiral where the hydrogen bonds held the molecule together. This was the first idea of a single-stranded helical structure. In 1949, Aaron Chargaff was the first biochemist who measured the nucleotide amount in DNA and refuted the tetranucleotide theory. He isolated DNA from different organisms and measured the amount of the 4 nitrogenous bases. His results concluded there were very similar amounts of adenosine to thymine and very similar amounts of guanine to cytosine. This discovery was an important clue in Watson and Crick's discovery. Using the technique of X-ray diffraction, Rosalind Franklin shot X-rays through DNA crystals to get a 3D picture and confirmed the helical structure of DNA. Wilkins then passed these images to Watson and Crick. Meanwhile, Watson and Crick were trying to figure out the 4-piece puzzle of the bases A, T, G and C, as discovered by Aaron Chargaff. They tried all the possible unions between A, T, G and C and one was perfect. 2 bonds between A and T and 3 bonds between G and C fit in a double helix. The 4 units structured this way could code for a huge amount of data. They submitted 2 papers where they only hinted of Franklin's contribution, however. In 1962, Watson, Crick and Wilkins won the Nobel Prize for their contribution in the discovery of the DNA double helix. However, there is a rule in which you must be living to receive the award, and Franklin had since passed at the age of 37 with ovarian cancer, which very possibly might have been from her overexposure to radiation from her research.
So now we're going to look at the physical structure of deoxyribonucleic acid, better known as DNA. DNA is, as we have just seen, a double-stranded molecule, from which all the information that is used to make us who we are comes from. Now, to really see how the structure of DNA is constructed, we need to unwind the 2 strands of DNA and separate them. When we do separate the 2 strands, we can see the basic components for each are virtually identical, but they run in opposite directions. We'll hear more about this later. Each strand of DNA consists of many nucleotides bound together by phosphodiester bonds to form long chains called polynucleotides. I know, a lot of long words there, but by the end of this video they will all make sense, don't worry. Now, what is a nucleotide? A nucleotide is one of the structural building blocks of both DNA and RNA. A nucleotide has 3 components. A 5-carbon sugar molecule called deoxyribose. This is very similar to RNA's sugar molecule, ribose. However, this ribose molecule is missing a hydroxyl group on its 2' carbon. A nucleotide also consists of a phosphate group and one of the 4 nitrogenous bases, adenine, thymine, guanine, and cytosine. So every nucleotide in DNA, there are many, differ only in which of the 4 bases are attached. How all these components are arranged is very important for how each of the polynucleotides are formed. A phosphate group is bound to the deoxyribose sugar molecule at either the sugar's 5' carbon or 3' carbon. The numbering of these carbons is essential in describing the directionality of the DNA strand. This same phosphate is also bound to another sugar molecule next to it at the alternative 3' or 5' carbon of the sugar molecule. This bonding of sugars and phosphates is what forms the phosphodiester bonds I mentioned earlier. You might be wondering, how do the 2 strands of DNA connect? How do they interact? And the answer is all down to 2 words: base pairing. So, the 4 nitrogenous bases mentioned earlier, adenine, guanine, cytosine, and thymine, can be split up into 2 groups. Thymine and cytosine are pyrimidines, and adenine and guanine are purines. When bases pair with other bases from another DNA strand, they have specific partners. Pyrimidines always pair with purines, and we can go even further to say that specific pyrimidines pair with specific purines. In the case of DNA, cytosine pairs with guanine and thymine pairs with adenine. These bases pair by a type of intermolecular bond that we are all too familiar with, hydrogen bonds. It's through these hydrogen bonds created through base pairing that the two strands of DNA are joined. As well as hydrogen bonds, DNA also has pi bonds. These help to form the double helix structure that DNA is so famous for. The sharing of electron probability, created by these pi bonds, creates two different types of grooves in the structure of the double helix, the major and the minor grooves. The grooves act as binding sites for incoming proteins, needed for DNA reading and replication. Proteins know which grooves to bind to by recognition of specific base sequences present in these grooves.
I will be talking about the chemical structure of DNA and how we classify their bases. As said earlier, nucleotides are composed of three sections: the phosphate backbone, a sugar, and a base. The first section, the sugar, is a monosaccharide, which is a deoxyribose, meaning we've no hydroxyl group attached to it. Extending from the anomeric carbon is a heterocyclic base. The base is attached by a beta-glycosidic link, which is a type of covalent bond that joins a carbohydrate to another group. It is a heterocycle as it is a ring structure with at least two other elements as members of the ring. In this case, we have nitrogen and carbon. It is also a base because it has nitrogen atoms with lone pairs. I'll go into this in more detail on the next slides. These are both attached to the phosphate backbone. The phosphate is attached to the carbon 5 of the sugar component. These together make a nucleotide. Nucleotides are the building blocks of what forms nucleic acid, which includes DNA and RNA. Next, we can look at how strands join together to form our double-stranded helix. Bases are joined together by hydrogen bonds. These nitrogenous bases are classified into two different groups: purines and pyrimidines. Purines are double ring structures, and they include adenine and guanine, also known as A and G for simplicity reasons. Pyrimidines are single ring structures, and they include thymine and cytosine, also known as T and C. Only a purine can join to a pyrimidine. Adenine joins to thymine, forming 2 hydrogen bonds, and guanine joins to cytosine, forming 3 hydrogen bonds. As we can see, the bonds are between oxygen and nitrogen and hydrogen atoms. Next, there is about 3 billion base pairs per cell in the body. If all the DNA that is in one cell is stretched out, it would be around 2 meters. If the DNA in every cell in the adult human body was stretched out, it would cover the diameter of our solar system twice. Histones are key components on how our DNA is organized and made into the compact structure of our chromosomes. A histone octamer is an 8-protein complex found at the center of our nucleosome core particle. It consists of 2 copies of each of the 4 core histone proteins: H2A, H2B, H3, and H4. 165 base pairs wrap around each octamer 1.65 times. A chromatosome consists of a nucleosome and a H1 histone. A H1 protein binds to the linker DNA region between 2 nucleosomes. Histones are positively charged. This is due to lysine and arginine, 2 amino acids found in the protein. They bind to the negatively charged phosphate backbone, forming salt bridges and hydrogen bonds. The nucleosomes are then wrapped into a 30 nanometer spiral called a solenoid, which then form loops that have an average length of 300 nanometers. These loops are compressed and folded into 250 nanometer wide fibers. They are then tightly coiled to form the chromatid of our chromosomes. This extensive wrapping and coiling procedure is how all our DNA can be packed into the nucleus of every cell in our body.
As mentioned previously, DNA is double-stranded. Base pairing in DNA is highly specific. One purine and one pyrimidine will fit perfectly together between the 2 strands. A cytosine can only pair with a guanine on the complementary strand, while an adenine can only pair with a thymine on the complementary strand. This specific pairing is down to the hydrogen bonding between the bases, where an adenine-thymine pair makes 2 hydrogen bonds and a cytosine-guanine pair makes 3 hydrogen bonds. No purine-purine or pyrimidine-pyrimidine pairs can occur. This is because the distance is too long for hydrogen bonds to form between 2 pyrimidines and too short for 2 purines in the helix. The exact order of these base pairs make up the unique genetic code. If a base pair is deleted, inserted, or inverted, mutations can arise. This will cause our genes to lose function and can result in genetic disorders such as cystic fibrosis or haemophilia, for example. It is also known that random errors in DNA can account for 2/3 of cancer mutations, so incorrect pairing can be fatal. Looking back at our DNA molecule, the 2 polynucleotide strands of DNA are said to be antiparallel to one another, with the bases projecting inwards. The phosphate backbones are seen to be upside down in relation to each other. One strand runs from what is called the 5' to the 3' direction. This means the first phosphate is connected to the sugar molecule's 5th carbon, and the next phosphate will go ending at the 3' end of the sugar. The complementary strand will go from the 3' to the 5' direction, where we get the exact opposite. We can now see antiparallel means the strands run in opposite directions parallel to one another. These strands twist in a complete DNA structure, giving us the double helix. This last section in the video will go into detail about the conformations of DNA structure. The most common form of DNA is in the Watson-Crick model, known as B-form DNA, which has been discussed thoroughly throughout this video. However, under certain conditions, other forms can be found. For example, aDNA and zDNA. Here is our typical structure of bDNA. To recap, bDNA is a right-handed helix with the planes of nucleotide bases being perpendicular to the central axis. There is a major groove and a minor groove. One complete turn is 34 angstroms and there are 10 base pairs per turn. This is the most common form of DNA found in organisms. Next, we will examine aDNA. Similar to bDNA, aDNA is a right-handed helix. Compared to bDNA, the major groove is wider and deep while the minor groove is more narrow. Base pairs per turn is 11 and rise per base pair is 2.3 angstroms. This form of DNA will only occur in dehydrated samples. aDNA and bDNA were both identified by Rosalind Franklin by X-ray diffraction analysis, as mentioned earlier in this video. aDNA's helical conformation also occurs in double-stranded RNAs. Finally, we have the structure of zDNA. zDNA is a left-handed double helix and is long and thin compared to bDNA and aDNA. There are 12 base pairs per turn and there is only one deep helical groove. The phosphate backbone winds in a zigzag fashion. The purines have syn conformation, meaning the base and the sugar are on the same side, while the pyrimidines have anti-conformation, meaning the base and the sugar are on opposite sides. This is a unique characteristic that zDNA has, where adjacent sugars have alternating orientation, unlike A and B DNA, which have the same orientation. Here is a table that summarizes the differences between the 3 conformations of DNA.